What if I told you that your birth certificate is lying to you? While your chronological age only goes up, your biological age can actually go down. For decades, we viewed aging as an inevitable decline. But the latest breakthroughs in clinical research are revealing a different story. It's not just about the passing of time, it's about cellular integrity. Today, we're moving beyond traditional medicine to explore how we can reprogram our biology at its core. In this video, we are diving into the mechanisms that govern your health span. Backed by the most rigorous data available in 2026. Let's begin. According to recent studies on PubMed, the key isn't a magic pill. It's a process called autophagy. Think of it as your body's internal recycling program. When activated, your cells literally eat their own damaged components to regenerate. This process is vital for preventing the protein buildup that leads to cellular aging and chronic inflammation. In 2026, we now understand that autophagy is the cornerstone of metabolic flexibility and longevity. The most effective way to trigger this cellular cleanup, it's not a supplement, it's time. Data from the Mayo Clinic suggests that time-restricted feeding is the primary trigger for this process. But how does this cellular cleanup translate to our most vital organ, the heart? Cardiovascular aging is often defined by vascular stiffness and the accumulation of arterial plaque. By monitoring biomarkers like high-sensitivity troponin, we can detect cellular stress before clinical symptoms appear. Standard tests often miss early vascular changes. True longevity requires looking at the endothelium, the inner lining of your heart. As we age, arterial stiffness increases, but research shows that cellular repair mechanisms can maintain vascular elasticity. Clinical data from leading institutions suggests that these processes are closely linked to our daily lifestyle choices. On PubMed, hundreds of peer-reviewed papers confirm the impact of time-restricted protocols on cardiac biomarkers. But what fuels this repair? To find the answer, we must go even deeper, to the engines of the cell. Mitochondria are more than just energy producers. They are the regulators of cellular life and death. When mitochondria fail, reactive oxygen species increase, causing cumulative damage to our genetic code. In the field of oncology, this genomic instability is recognized as a hallmark of early-stage carcinogenesis. Hematological studies are now exploring how healthy mitochondria can support hematopoietic stem cell. To combat this decline, we must look at how we fuel our systems through metabolic switching. Metabolic switching involves transitioning from glucose-based energy to the consumption of ketones. This metabolic shift serves as a primary stimulus for cellular autophagy and mitochondrial biogenesis. In this state, sirtuin proteins are activated, directly mending strands of damaged DNA. The core fuel for these sirtuins is a coenzyme called NAD+, which declines as we age. Current clinical trials are examining precursors that can effectively replenish our cellular NAD+ levels. The synergy between nutritional timing and cellular repair is the foundation of modern longevity medicine. Intermittent fasting, specifically the 16-8 protocol, remains the most studied intervention for autophagy induction. Clinical observations at the Mayo Clinic have highlighted the impact of these protocols on systemic inflammation markers. In cardiology, these interventions are associated with improved left ventricular function and reduced oxidative stress. However, the effectiveness of these protocols is highly dependent on individual biological responses. In 2026, wearable technology allows us to track real-time biomarkers, from heart rate variability to glucose spikes. Epigenetic clocks represent the gold standard for measuring our rate of biological aging at the DNA level. Without precise measurement, any intervention is merely guesswork. Data is the bridge to true longevity. We rely on peer-reviewed literature to distinguish between fleeting health trends and validated clinical protocols.
Ultimately, understanding our biology is the first step toward mastering our time.
Deep repair is not possible without optimized sleep architecture. It is the time when the glymphatic system flushes neural waste. While the market is flooded with supplements, clinical longevity prioritizes substances with proven efficacy in human trials. In hematology, tracking C-reactive protein levels provides a window into systemic inflammation that drives chronic disease. Artificial intelligence is now being integrated into clinical research to predict biological age with unprecedented accuracy. The shift from reactive to preventive medicine is not just a trend, it is the most cost-effective investment in human capital. DNA methylation patterns act as the software of our cells, determining which genes are expressed and which are silenced. For cardiac patients, personalized longevity protocols can significantly reduce the risk of secondary events by optimizing lipid profiles. International classifications are beginning to recognize aging itself as a modifiable disease state, opening doors for new therapies. Building your longevity roadmap starts with understanding your current baseline through rigorous clinical testing.
We are now 7 minutes into our deep dive. The next 3 minutes will cover the most actionable steps for your biological future. Longevity is driven by hormesis, the biological principle where brief exposure to stress makes your cells stronger. High-intensity interval training is a primary hormetic stressor, optimizing cardiac output and vascular resilience.
However, as we age, some cells stop dividing but don't die. These are known as senescent or zombie cells. Oncology research is currently exploring senolytics, compounds designed to clear these zombie cells and rejuvenate tissues. By clearing cellular debris, we can potentially delay the onset of age-related cardiovascular and neurological decline. We cannot ignore the role of chronic stress. Elevated cortisol levels are a direct catalyst for telomere shortening. Nutritional oncology suggests that specific polyphenols can mimic fasting effects, activating our longevity genes. Large-scale clinical datasets are the only way to validate which protocols actually move the needle on human health span. Transitioning from a patient mindset to an optimization mindset is the final frontier in longevity science. We have covered the mechanics, the protocols, and the data. Now let's synthesize this into a daily roadmap. Step 1. Prioritize your feeding window. A 16-8 protocol is the most accessible way to initiate daily autophagy. Step 2. Master your recovery. Deep sleep is the non-negotiable phase for systemic, cellular, and neurological repair. Step 3. Consider evidence-based supplementation. Boosting NAD+ levels supports the enzymes that repair our genetic integrity. Step 4. Monitor your cardiac health. Use data to track vascular elasticity and manage systemic inflammation. Remember, protocols must be personalized through clinical testing and professional research review. We are at the dawn of a new era where aging is no longer an invisible enemy, but a manageable condition. At Clinical Longevity, our mission is to decode this science and bring the latest data directly to you. Don't just age, optimize, because your biological potential is the ultimate asset of your life. Subscribe to stay at the forefront of the Healthspan revolution. Thank you for watching.